So basically, I titled my talk like how I got into this virology program and where I am like right now. So of course, like in the big from the very beginning, I was like interested in science, and I would wanted to do. Actually, I wanted to be a doctor, like a medicine, like an MD doctor. So I was in India. So I did my undergrad back in India, and in India, after while you're preparing, like in high school itself, you need to basically pick up whether you want to go in science background, like you want to choose your biology, or you want to go towards the engineering side. So I was in the biology major, and I had like maths, physics, chemistry. So I took that route because I was wanted to be an MD doctor and a clinician basically. But I have so just so happen around the way that after the high school, you basically have to sit for this board exams, and then like it's the entrance exam for medical schools. And all my life I've been in Delhi in India, and I didn't know at that point I didn't want to like go outside. So I got into medical school, but the medical school was not in Delhi. So at the same time, I sat in like so many entrance exams, but I got into this engineering program, which was basically a marriage between science and like engineering. It was in biotechnology. And one of the things that really fascinated to me about biotechnology was that you are still studying like very close to science. You has like maths. I was like really, I really love like doing quants and like calculus and all that. So I was like, okay, so this is a good start, and I still can be in like the science degree. I can still pursue. I can, if I want to, I can still go for a PhD, MD, PhD program later in life. So that's basically what stated from my high school to getting into a professional degree of getting like a like engineering degree. But so that being said, when I actually did my engineering, at that point, I had like no idea what I'm going to do next. I had like some seniors along the way, and they were preparing to come to grad schools in US. And I'm like, okay, so I will apply because, it's always helpful to have like good mentors and like, the seniors whom you can talk to and like get some career advice. So I applied to multiple schools, grad schools in US, and I got admission. And that's like the first time in 2005. Um, oops, cannot, that I took my flight coming to US. And that was back in 2005. I never sat in plane because I was always in like a Delhi person. I never left my city. I stayed with my parents. And when I was 21, I left and I came to US to pursue my graduate degree because I was just interested in learning more about science. And at that point, it was like mostly like a general science, not like uh, like I didn't know what I really wanted to do because most of the time in the grad school, when you're applying, you need to pick up like a major. But I didn't know. I mean, I was interested in general science, and I had an idea, I wanted to do more cancer research, but the virology didn't click. Even in my undergrad, we didn't even have a virology 101. We had microbiology. We learned about bacteria stuff, but nothing about virology or viruses. So that was me taking that plunge. But the reason why I came to US and I joined Penn State for my grad school, because it was like this integrated bioscience program. So this basically lays out like my basic path that I have from where I started and where I am here in like 2021. So like I said, like in 2005, did my undergrad in biotechnology. In 2011, so I came here in 2005 itself to pursue my graduate degree, and then the graduate degree was completely very new. Like, and then also the one of the things that completely struck, like the studying from India to US is completely a different thing. Here, like you learn more practical, you have like more lab work, and in the undergrad, I had like no lab experience like whatsoever. It's only like my final year we were supposed to do this small project, and I was involved in like a lab oriented project oriented research work. And that really kind of paved my way that I really am interested in applying to the grad school. But at that point, I still wasn't sure which major I'm going to choose. So I kind of tried applying to like umbrella programs. So one of the programs that I applied, which I got admission into Penn State, was integrated biosciences. And it basically gives you this opportunity to rotate in various different labs from different departments. So you were not like, you don't have to really choose the lab while you apply. You basically get the opportunity to rotate to different rotation, different labs. And that kind of really helped me to understand what I'm really interested in. So I took that first year rotating in multiple different labs from immunology to cell development biology. And then finally I met like the things that I really got like fascinated about. And that was my graduate school lab was in like in the in that was studying viruses. So at that point, like I said, I did not know much about viruses. But what really fascinated to me was, like these tiny little things like cause so much damage. And then what we were trying to study, the lab that I was rotating, they was trying to study like how the viruses can cause like damage to the host cells and how we can combat that. Like, can we understand like the different intricacies between the virus and the host interactions and how they actually regulate your immune system? So all these questions were very fascinating, and these tiny little like pathogens causing so much damage. So I was like very much enthralled in my first um, acquaintance with like studying viruses and just like working in the lab with like with viruses where you can do like more quantification stuff. There was like the different assays that you can do to quantify it, the virus, and like it was really nice. And the other thing that really helped me was my PhD advisor, who is like my great mentor that helped me through my career. He was working in the lab with me at that time, and that really helped me understand exactly like the techniques that you do, from the cloning to running a basic like Western blot gel to actually doing infections with like the virus, growing the virus. So all those things I learned along the way. So of course, like the journey, like you have to put in like a lot of hard work. So that was not the problem. It's just because I was like so interested in what I was doing, I didn't mind that at all. And that was just like the start. And then going forward, I took classes and coursework to understand and get my grasp over virology, over immunology. And after I finished my graduate school in 2011, I joined my postdoctoral work at Icahn School of Medicine, Mount Sinai, which is in New York. And here I was in microbiology department. So in my grad school, I studied RNA viruses, a different kind of, so there are multiple different RNA viruses, and of course, right now, the world knows more about viruses than they ever did. So I don't want to give you too much introduction, but of course, the viruses that I study, of course, they don't cause that much lethal disease, but I was studying the family of viruses that belong to the mumps and measles, which you guys have already heard of when and in the child when you get this MMR vaccine. So in my postdoctoral work, I actually went from a higher risk pathogen viruses. They are called like BS. So for the viruses, there are different level of the biosafety level. So 4 is considered the highest, and as you guys know, SARS-CoV-2, which is now causing like such a havoc and a pandemic, is like a BSL level 3. So I started working in postdoctoral work on BSL level 4 viruses, which are the filoviruses that includes Ebola and Marburg. And then one of the things, of course, in New York, we don't have like, so in order to work with these viruses, you need like high contained labs. You just cannot work in like the regular lab with the biosafety cabinet. These are like very much highly sophisticated engineered with like the right kind of air pressure, to have negative air pressure, and you wear proper PPE, which involves wearing like a proper hazmat, like a suit, which has like a proper respiratory protection, um, gloves, gowns, and like Tyvek suits and the booty. So you have like a completely different ball game when you are working with these pathogens. But however, with the BSL-4 at that time, we didn't have the BSL-4 in New York. So what we really did was looking at, because these viruses are made of like RNA, DNA, and proteins. So the viruses that I work with is they are the RNA viruses, and I was interested in understanding how the viral proteins interact with the host protein. And one particular protein was that is important for the virus to evade immune responses. So that was like my first kind of like get go of like getting started with this highly pathogen virus. And of course, because in academia or any research work involves a lot of collaboration, so we made a lot of collaborators, and our collaborators who had these highly sophisticated BSL-4 labs. So if you find something in cells that makes sense, and we want to see if that could decrease the virus growth or has an effect, we can always give that to our collaborators, and they will test it for us with the actual real virus. So for the long time, that's how like the work day began. And from the postdoctoral fellow in 2013, I was promoted to be instructor, and I involved in like some teaching in the department, where I also had exposure to teaching like medical students as well as grad students. So it was kind of really nice. And we talked about the medical microbiology. And of course, in addition, we did the research. Then in 2016, I moved to Georgia State as a research assistant professor. I continued working with filoviruses at that time. And one of the fascinating thing about Georgia State was that we were going to be having our own ESL4 labs, and that was kind of really interesting to me because all these years I've worked for the virus, understanding the molecular intricacies of like how virus protein interact with the host proteins, but I really didn't have the experience of working with the actual pathogen. And this was the opportunity because Georgia State had the BSL4 labs. So while I did that, the other areas that we've been interested in, like understanding once we identify some pathways or some targets, how we can actually develop them to get to the therapeutics. Because at the end of the day, you of course want to understand how a virus is replicating, or you want to find like the weaknesses in the virus part, like you can actually stop it from growing therapeutically. So the approach that we have been like working is like thinking about how to understand deeply about these mechanisms that can help us to design better therapeutics to suppress these viruses. And then because I'm not sure how familiar you are, so academia and non-academic, there are two different routes that you can take. So once you're a research assistant professor, which really means that basically I don't have my own lab, but I had like my director of the institute, who was also my boss, like he has like the big lab, and under his umbrella I was getting paid. But of course I had the opportunity to write my own grants, but at that time I was also seeking for my own independent position, where I can have my own lab, I can have my own students and postdocs and the research staff, and actually stand my own research group to study the things that I would like to continue on. And that's where I landed up as a principal investigator last year in Trudeau, right when New York State went into the lockdown. So it was kind of interesting story that happened. So when I interviewed here, of course, like the theme was the same, that I have to stand with like the virology core, or working and establishing a virology lab with the focus on the BSL-3 pathogen, because at Trudeau Institute, we have like really fascinating and amazing BSL-3 facilities. So the for me, the goal was to actually start a research group focusing on emerging viruses, and one of the viruses family that were going to focus on was the tick-borne viruses, because tick-borne viruses are kind of endemic to this region. But right at the moment, we were right in the middle of pandemic, and the call of the action was SARS-CoV-2. So very quickly, the goals and like the whole attention kind of diverted into the SARS-CoV-2 two direction, about which I'm going to talk to you a little bit more in few minutes. But just to give you give you a background of like what Trudeau Institute is and where we are. So So I'm not sure how familiar you are. So if you look at New York State, it's pretty big. So and the point over here on the, let's see, New York City is marked like right here. So we are right on like upstate New York. We are almost like close to the Canadian border. Like Canadian border is like an hour away, and like the closest biggest city is Montreal. So we're situated in this like nicely like a nature centric area surrounded by lakes and like mountains. So it's really nice. And Trudeau itself is situated on the lake. It's called Saranac Lake. And one of the key history about Trudeau Institute itself is that, so this institute was started as like the first American laboratory to study TB. So Edward Livingston Trudeau, he was a doctor back in 1800 when there was no antibiotics for TB. He got sick with TB and he came up in the mountains that he's like, he's going to get the fresh air and, he's going to live his end days and then, end his end of the days here. But what happened was when he got here with TB, he actually started getting better and he thought like maybe there's something in the air of this place that making him better. So he kind of dedicated his life and opened this institute. He had like these cure cottages for the people who were suffering from TB before the antibiotics came in, and we still have one of the cure cottages. It's the model right in front of the institute. This was like one of the snowy days that we had, and I mean it's cold in upstate New York. So this is like where like people who were sick with TB, they would stay here, and inside here we have like kind of like a small museum around on campus, and which basically it has like a patient bed and then shows you inside. But it is kind of like very um, fascinating about this place, and one of the reasons I chose here, given its strong history of curing infectious diseases. And that's where the challenge came in when I came here and I joined, and when we were hitting by the SARS-CoV-2, we had no time to waste, so we jumped right into it just because we could. We had like a BSL-3 trained staff because SARS-CoV-2 is a highly pathogenic virus and it requires very professional work with respiratory protection, and it's not BSL-4 like Ebola or Marburg, but it is BSL level 3. Still requires a lot of like high contain like the PPE and then respiratory protection to be working in this sophisticated lab with all the engineering controls. So these are just like the pictures that we share. So this is the lake on the campus for that was in the summer, and like right now this lake is frozen. It's still very pretty. It's cold, but it's really really pretty. So I find myself, like in the lockdown coming up, we were given this opportunity to gather a team together and start working for the SARS-CoV-2. And like one of the works that my lab is like currently doing is uh, to basically is to develop a drug discovery and the biology program to identify therapeutics for SARS-CoV-2. So we have our inbuilt small molecule screening for the drug discovery platform. And also we work with partners who have their drugs that we could test against the virus, because not everyone has access to BSL-3 labs and like the trained staff. So in that way, so we started working. So of course the last year was like super busy getting the team together, people together, getting training and getting all the stuff that we didn't know about the viruses and like establish all these assays. So it's been like a busy 2020, and starting 2021 has been like almost similar. So that was like a little, a very overview of like how I started and how I ended up. But talking about my love for viruses and like, why would someone study virology? For me, specifically, like I said, I was like really fascinated by these tiny little pathogens who have such a potential, who can transmit, get into the host, cause disease, and they have like very smart ways of evading immune responses. Like, they even the host is there, they can find it. They're very sneaky and they can find their way to control the host. So the question becomes is like, how do they do it? And that's like the basic question of understanding how do we tackle it. And of course, there are a lot of sophistications involved. And starting from this simple question, that's exactly how my interest kind of got intrigued about this virus and like the host interaction. And for all this time, from starting from grad school and up till now, I've been like trying to picturize this big summary of like, how I think like the virus and host are always like as at in a tug of war, where the virus is coming in the host system, where they're trying to overcome the host immune response, and the host will try to like subside the virus growth. So both, and then there is a fine balance between them. The only once the balance is tipped from one way to the other, that's when like you get into the sickest stage, you cause disease. So every virus have this own fine tuning with the host to interact with it and to cause the disease. And as a virologist, my goal is to understand how they do it. There can we look at the fine tuning of these protein-protein interaction? Can we find some pathways that we could target for drug discovery? And then, of course, from the basic research gets into the translational point, and if we can find those targets, and if they are druggable targets, that's where the therapeutics come into play. And of course, this is all starts with like from this very small scale from the cells, and then it goes into the animal, and then into the clinical trials, and then finally to the patients. And then the vaccine route is also the same. And the vaccine thing is like you want to stop the infection, uh, stop the virus infection, and that's why like both the vaccines and the therapeutics are needed to combat completely these viruses. And then here's also throughout my journey that I've studied BSL2, BSL3, and BSL4 pathogens. And right now, my whole world is surrounding SARS-CoV-2. Can we help out coming up with like better targets, better therapeutics, and can we get like the drugs to clinical trials? So that's what my group is like hoping to achieve in this like small span of time, of course. And then, the one thing that has really worked and helped me to get through and then assemble this team is really the scientific community. People have been like so helpful sharing reagent. The science has advanced like in such a tremendous speed because everyone is working with each other. There's like no hesitation. People are working day and night. Like even my lab, considering we have worked, you know 20, there have been continuous days of like 20 hours work, and then the work never gets over. Even if you're done in the lab, you still have to write, and you still have to complete the progress, the reports, and like you still have to write papers. So the work is like never ending, but at the end of the day, the reward is getting something out to help people. And I think that's is like the most like driving factor for me at and that's always been for me that has stuck me into this career, and I really love what I do. So, like this is just the background about the science, but in general, like a day-to-day life of a scientist is not like as simple as it sounds like. So I just put like a simple pie chart over here. So even though I have my lab, I have like 4 people who work with me, I still have a lot of lab work to do. So the 70% of the lab work, which means basically you have to design the experiments, you need to think about what the next steps are. You actually are actually running the experiments both in the BSL2, BSL3 labs, and whenever you step into the BSL3, which is the high containment lab, the time goes like really slowly. Like, I mean, or I should say, time goes really fast because you have to work really methodically and very, very patiently. So, if anything that in a normal, like a BSL2, like a biosafe BSL2 lab would take you 1 hour, in the BSL3, the same amount of work will take you 3 hours, just because you're going at a very slow and pace. So you want to make sure that everything is contained. You have to decon everything. So it's very methodical work that goes on in BSL3. And of course, people who have had like experience of working in the lab in BSL2 at least for several years, only after that they're trained to get into the BSL3 labs. And that's what we have a training program in BILS. So my staff got trained here, even I, because no matter which institute, even if you had a prior training, you still have to retrain in whenever you join a new institute. So that's all takes like a lot of time and effort, and of course a very coordinated effort. But other than that, this lab work, of course, there's a lot of writing, and the writing means you have to write, apply for grants. You have to get money to do research. We have collaborators, so we have to write, the papers together. So, for example, for SARS-CoV-2, as we started just last year, it's been, and there have already been like so many papers out there. So the scientific community need to know what you find so that they can move forward. They can make the next steps, move forward quickly. So right now the science is going at the speed that I've never seen. Previously we would like work on it, manuscript would take several years, but right now the manuscripts are like coming in like months time. So that would tell like how much time that people are spending in the lab, outside the lab, just working together. And then it's not like individual, it's like very collaborative. And even within my group, we, five of us, we work very, uh, like a lot of this time we work uh, in a very coordinated manner. But of course you need help with like writing grants, and then other PIs, so we write grants together to submit because we need to get money. And also they're like, then there are collaborators outside the institute that are also interested. So you have to keep track of all these things. And then because I am also play a managerial role, because I have people to manage, so I have to spend some time doing that as well. And then there are like a lot of meetings, internal meetings, we have group meetings. um, we have the discussion about like we finished project A, now we have to move on to project B if there's some troubleshooting needs to be done. So all that takes a lot of time. So one thing that is really really important, no matter what level you are, you need a really good time management. Like no one can like stress that more. I think something that you need to put that into your practice. One thing that I've started doing, no matter, even if it is like the smallest task, or even if it is a 15-minute thing, I put it on my calendar, because my calendar knows exactly what I'm doing. Of course, sometimes you can't completely follow that route, but all it helps is to organize yourself for your day, for the week. So in that slot, you already know what you have done. So for example, if I'm going into BSL3, I know I'm going to be there for 5 hours. So that means my chunk of my like big day is gone inside the BSL3. And in the BSL3, I can check my emails when I have like not working in the lab, in the if when I'm not working with the pathogen, and I'm waiting or incubating, waiting for stuff to incubate. But I really cannot like come out. I mean, you can come out, but like you have to give yourself enough time. So it's not like you can do frequent back and forth that much, because you still have to, doff your PPE, and you go out, change, and then come back. So that takes like by itself another 30-40 minutes. So you have to plan your day really really well. So I have to plan my meetings around my schedule for that day if I'm going into BSL3, which was because today was the same thing. I was like in the suite from like noon to like 5, and then I'm here talking to you guys right now. So, so that you need to have like really good time management. Of course, it takes like a lot of effort, but one thing that will really help you engage and, find your interest, if you are interested or you love something about it, you will be passionate. So the passion is what drives you. And I think for me, the science and like doing experiments is fascinating, and it drives me every day, because I would like to know what is happening. If I'm running this experiment, what does this drug block the virus or not? Do we have like a path forward? So I'm like always like inquisitive about like what's going on, and that like really really gives me, keeps me driving the science that I do. But more and more, my lab gets established, I think I kind of like have to like step away from like the bench work a little bit, just so that I can take care of the other things, because of course if I'm working in the lab, it's very hard to find time to write grants, and grants are important, like I said, because you have to get money. So that's like a very typical, like an academic setting, because I'm like a new PI. I still have like a lot of hats that I wear. I mean, like I said, I'm like still doing research in the lab. I'm doing experiments in the lab, running like planning the studies thing, writing. So, but I think more of my lab members start getting like a more familiar with the work that we do. I think part of that work, so I can step away from the lab and concentrate more on writing and taking care of the other administrative work, which I have not been doing as much as I could. Trudeau Institute is like an independent research institute. We do not have graduate students, but we do have a partnership with like a local, like a Clarkson University, and we get undergrad. So, for example, because last year was kind of subdued because of pandemic and lockdowns and everything going online. But this year we finally have like students who are doing some lab work. So those students will actually get come together for summer. So they come in the spring semester, and then they come in like the summer semester, to do proper research work. So that's when like I'm gonna get some the undergrad working in the lab, who would like to learn basic techniques in BSL2, and then the program will be giving them like a small project so that they can finish in span of the summer. So that will be kind of exciting, because back in both in Georgia State and Sinai, I was involved in teaching, so I like a little bit of that too. But once the teaching involves, you can see the lab work will going to decrease. But again, like I still have to look at over the data. There are a lot of things that you still have to do beyond the lab, because you analyze the data and then you put the papers together, manuscripts, submissions, and then the grant submissions, and they're always like a deadline that you're always working with. So like I said, one of the key things that really helps is like keeping track of your time, and setting your milestones and goals for the year, for the semester is always helpful. So so that was like an academic career that what I am pursuing. But of course, if you have a science degree, you can do so many things. And of course, that would mean that you would probably want to go to grad school. It doesn't matter what major that you choose. And of course, if you have graduate degree in infectious diseases, you have like multiple paths that you can take. Of course, public health remains like one of the key things. And then the public health, you can be an epidemiologist. You can actually figure out the infection, then figure out the transmission, what we are doing seeing with the SARS-CoV-2, like how the virus is spreading. They're actually keeping track of like the patient, so you're working on the patients and on the virus where the actual outbreak is happening. And I know some of my former colleagues, they have gone into that route, and they just love it. And of course, like I said, you can always opt for a medical school with the MD PhD program. And that way, of course, the years will be longer, but you will be able to, in addition to have your clinical experience, you will also have like a lab background. So you can actually contribute a lot more um, to the science and like to the critical questions that you ask. For us, when I have to partner, I have a clinical question, I have to partner with like a clinical um, lab, which is not a problem at all, because people collaborate in science. But if you are already an MD PhD, it opens up like more opportunities. But again, you can either be a clinician or both, have a research lab as well as clinical lab. And then, the other career options is, of course, going to pharma and biotech. I mean, right now, the field is like massive. Like they're always looking for scientist position, even postdoctoral fellowships that you can do in pharma and biotech. And other than that, I mean, I have friends who have had a PhD in microbiology doing 2B research, but she's right now a science writer. There are students that people that I know, they can also go in consulting after they're getting a PhD in science. Um, a few friends who became editor in like big science journals. So still, the options are like various. So it all depends like what you're interested in. Of course, these are the things when you are not super interested in doing lab work, but in pharma and biotech, it may still require, depending on where you are kind of entering, still may require to do some of the bench work. But I think eventually, as you move along, you step away from doing bench work. So, I mean, having like a degree in science opens up a lot of options. And of course, you need to find what is your passion first and what interests you first. And then figure out your major. And then, of course, like going along the way, you really need to talk to the right people. If you're interested in medical field, you need to find people who are doing research. There's like for the undergrad, you need like shadowing experience. Same for the graduate school. I mean, I didn't have much of like the the work like work experience in the lab, but I learned everything in the graduate school. So it all depends, how willing you are, and like, how you portray your CV and where you see yourself going into for the next step, be it be graduate school, medical school, or and even if you choose like a wrong major or a graduate school, it doesn't really mean that this is the end, because you always learn something. There's always something to learn. And then the one thing that you really pick up in graduate school is your analytical skills, and those are the skill set that is really really helpful no matter where you go, be it consultant or science writer. Because what they really people are looking in the PhD is the way, like how they look at the data, they analyze it, um, and then just like a very thorough and a comprehensive thinking to put towards a solution. So I think that's what you have to should be focusing on when you're looking for your next step in the career, be it the undergrad and the graduate studies. Like, what am I learning? So always be willing to learn new things, and it's always helpful to put as many new things that you can. And of course, you cannot go too overboard, because it gets like busy, so you need to pick and choose. So priority is important. Like, you need to prioritize things in your life, and of course it will happen eventually. Like I said, for my grad school, I had to rotate in like 5 different labs before I picked up the one lab that I chose and became my career. So and people switch. And I had a friend who she was a PhD student with me at Penn State, but then she later on, went ahead and did masters in statistics, because she wanted to switch from the lab work, and she was more interested in like the quant and like looking in the business side of things. So people do lots and lots of things. So you need to find your passion first, and that will going to help you drive it towards your next step. So this is me working in the BSL 3 with the virus, and this is like a typical like the BS like the biosafety cabinet that we have, and then that's like the this is the respiratory protection that we use. You cannot see the hose, but it is connected with like a, a battery powered thing that gives you a positive air pressure. So that means every time you're breathing in, you're having a positive airflow. So you don't have like infected cell air. And then of course you're like double glove with like the sleeves and like the gowns, and of course at the feet are they have shoes, and then they are covered with booties and stuff. But a typical day in the BSL-3, this is I worked with like the cell models and like tissue cultures, not with the animals yet. And then you have to be whatever you put it inside in the biosafety cabinet stays inside. Everything is decon, and the gloves are changed, and then you come out very clean. So you have to be very meticulous, very careful whenever you're working inside the things. Of course, this biosafety cabinet situates in a room, which is like our BSL-3 lab, and it has like special access. Not everyone can get it, and then not everyone can get in. So that's me. And then, the last few words, I have, for your career, like I said, you have to be persistent, you have to work hard, but more importantly, it's important that you work smart, right? You need to figure out what is the important questions, we figure out what are your priorities. And throughout, science makes you ask questions, so you do need to ask questions. And those are the questions that will help you cater to your next step, or the next research thing, or next career pathway. And of course, all this along, I couldn't been here if I didn't have the guidance and help from my mentors, my graduate school advisor, my other colleagues, my like the senior postdoc that we had, other faculties that I interacted with. So there were like so many people that I can't even like think of, that really helped me figuring out, when you're applying for grad school, when you're applying for even jobs, you need like so much input. You need to put together like a summary of like what your research is going to be like, what your lab is going to be doing when you going to be coming as a faculty. And all these things, you need people who are there, who can guide you, who can like read. So like I had faculty members read my summary statements, and like my application that I sent it to for my job application, just to give me feedback and really advise like where to go, like what to do, because there is always going to be hurdles. And even like right now, when I started my lab, I always go back to them and figure and ask them, how did you deal with, managing your staff? How did you hire your first postdoc? What was your priority? Like there's always going to be questions. So don't be afraid to have good established mentors in your life that will help you along the way to get through what you are at currently. And also, the good mentors always stay with you. Like my PhD advisor, I can call him anytime, and he'll answer my call. Even my graduate school advisor, I mean, it's been like tremendous. Of course, now over the years you become like more friends, because you have like that relationship changes. The dynamics is kind of a really different. But I think what you really need to do is like make sure that you have these people that help out. And of course, the other thing is like think about your friends, your colleagues. So you need to be a good ally to them, a strong ally, so that you spend so much time with them, especially in grad school when your lab work could be like 16 to 20 hours. You're spending a lot of time together, so they become your friends. So you need to be there for each other, something that is going to be really helpful in your career and also moving forward no matter where you go. So those all be the last like few tips that you should ponder and like start practicing, and then start implementing in your own life, so that you start now, and that will be stay with you for the rest of your career and rest of your life. So I think that was all me talking. Talking about, I might have skipped some of the things that I didn't talk about. So I would like to hear if any of you have particular questions that I could answer. Um, Virology, not virology, academia, not academia, anything in general that you would like me to talk about.
I think one question in the chat was what would be a BSL-1 virus?
So BSL-1, that's a good question. So the BSL-1 is not, basically viruses do not work at the BSL-1. So I would say, so the BSL-1, so the biosafety cabinet, it's like basically a hood which has like this laminar airflow. So any lab that has that containing that unit, the biosafety cabinet, it's considered BSL-2. For the virus work, just because they're pathogen, even if they are not caused like human disease, you still have to work in the biosafety cabinet. So the viruses themselves, any work is considered BSL-2, even though the virus might not be infectious. So the biosafety levels are basically defined by like how they cause how the viruses cause disease. Is there like any treatment available? If there are therapeutics or vaccines? So, and then of course how contagious that virus is. So in that case, so unfortunately with virus work, you always has to be at least BSL-2, unless it is like completely attenuated and completely inactivated with something. Like, you can always UV inactivate the virus and that will become a non-infectious. It won't replicate no matter if you put it cells. So one key things about virus is that they require a host. They cannot persist by themselves. They need something to go in to make more copies of themselves. So in that way, viruses are smart and kind of also dependent on us to give them the feed. So in the case of this lab, we don't we always work in a biosafety cabinet with the level 2, so to contain them, no matter if they're infectious to humans or not.
Can I ask a question?
Sure.
So, my name is Meher, and I think like your job as a virologist is really fascinating. And I was wondering, I know that you worked on Ebola. So, what was your experience like with that?
Yeah. So, I mean, Ebola was like fascinating. Like I said, I never got to work with actual infectious agent, which has always been the thing because I never had the chance to be in like a BSL-4 lab, except for when I went to GSU. Um, but one of the key things that really was cool about Ebola was, back in the day 2014, when like the big outbreak happened with Ebola in like DRC. At that time, we were already working with it, so we identified like uh, a host protein that could be targeted by the virus, and we were trying to see if can we therapeutically target that host protein in order to stop the virus from growth. And because we had such fascinating collaborators who had the BSL-4 labs, so we were able to very quickly, generate cells that they can actually infect with the virus, and we got data in like no time. But of course, this is like all very even before preclinicals. It's like basic research, like you go from the idea to the next thing. So that was just an idea that was tested on and it worked out really well. And then we worked more fully to understand, and later on we found that particular pathway was really important in containing even like the 2014 virus. So of course, and one of the things that really triggered from this work was like we started developing cell-based small molecule screening because for drug screening, and that was like I was really interested and fascinated about, just because um, the you know the drug screening would be like the next platform where you actually test the small molecules which have a potential of developing into something. And we did like big screenings with like our collaborators. I went myself, did a screen like over 400,000 compounds in the cellular base, and these are like more sophisticated test system where you have like robotics to plate the cells, because these are 384 well plates. And then lot of manual labor involved, but the bunch of like the addition of like the liquids and like the drugs are done by the robotics. So we developed that along the way, and I mean, yeah, it resulted in a bunch of papers, but unfortunately nothing to clinic yet. And that's the hope with SARS-CoV-2, that take it to the next level, get something out there for at least clinical studies. So that's the goal.
So, what were some of the differences bet- with like the epidemiology of like Ebola and COVID?
A lot. Very much, they're different. So to begin with, the first difference is, so both are RNA viruses, but Ebola is a negative strand RNA virus. That means the genome is kind of like in a reverse copy. I'm not sure how familiar you are with RNA DNA, but like our we are all made up of like the genetic materials DNA. The viruses could be have RNA or the DNA, but both these viruses have RNA as their genetic material. So the RNA in the case of Ebola is reverse, is negative strand. Instead of going for like A, A to Z, it goes Z to A. That's how it reads. But in case of SARS-CoV-2, it's a positive strand RNA virus. That means A to Z means A to Z. Um, so that's the first thing. Of course, there are two different family of viruses. SARS-CoV-2 belongs to coronaviruses. Ebola belongs to filoviruses. Both may have like origin from like bats. I mean, Ebola, like that's been like said, like the Ebola comes from bats, but of course for coronavirus, it could be like bats, pangolins, that's been studied. And in terms of like where, and of course, Ebola is, can only be spread. It's not like respiratory infection like SARS. Ebola is spread by bodily fluids. So that means you really need to be in close contact with someone who's infected with Ebola, and it's highly lethal. Like the case fatality rates is like 80 to 90% with Ebola versus with the SARS-CoV-2, the case fatality is still like less. I mean, it's more infectious and can spread faster than Ebola just because it's respiratory, it's in aerosols, you can spread easily. But if for Ebola to spread, you need to be in close contact with the person. But of course, the in 2014 that kind of changed because what was identified also in some patients, like it was found that the virus could be also sexually transmitted. It was found in the infected patient's semen sample for over a year. It was found in the conjunctiva in the eye. So a lot more new pathologies were like noticed in 2014 outbreak, which stands true for the new virus. But the SARS-CoV-2, on the other hand, is less lethal than Ebola, but on the other hand, it transmits fast. The transmission is like 50 to 70%, but the case fatality rate, even though it's spread so much, is is less. And and that's what is said because I mean, that's why if you look at the numbers, that we have so much more infections. And if you really think about the virus, a cool virus, well, a smart virus like SARS-CoV-2 is like it will going to go infect you, make you sick, but it won't kill you, so that you can go and spread, and so that it can go and like get to the another host. On the other hand, Ebola, if it gets into you, you basically, like most people die. I mean, right now we have like approved therapeutics and like there's the vaccine approved, but back in 2014 there was nothing. Like there were just experimental drugs, some experimental drugs. So, and then of course they were given the convalescent plasma from like the survivors to see if they can protect. But for the Ebola, like it kills. So if it kills, the spread of that infection doesn't happen as fast as like SARS-CoV-2. So they are completely different thing. And even SARS-CoV-2 from its own like ancestor, which is SARS-CoV-1, is very different in the same scenario. SARS-CoV-1, which happened 2003 in China, was not able to spread that much because it was highly lethal. So I think that's where make the SARS-CoV-2 a little tricky. And it's not only a respiratory infection. The other thing that we've been seeing, that it can cause infection in your heart, in your kidneys, in your intestines. And so it is, and it causes like a various amount of like the cascades within, it plays around with your immune system such how that it doesn't recognize, and later on cause like a very strong inflammatory response that actually comes down to being more lethal than protective. So it's a completely different beast than any of the other viruses that we understand right now, and we are still learning about SARS-CoV-2.
Yeah, thank you so much.
Yeah.
I think another question in the chat following up with your research on COVID-19 was, are you still working on it? And if you are, like how the experience has been?
Yeah. So that's, well, yeah, of course my group is working on it. I mean, like I said, the experience has been like fascinating. I mean, of course it was challenging because my lab started in the lockdown situation where we were in the emergency operations. So put a team together was hard, but thankfully we had really good trained technicians in the institute that where I was able to leverage their help and put like a group together, and we started working on like from the very get-go back starting in April actually. We got the virus very quickly. We grew it and then established the assays that will allow us to study the effects of different drugs. So we in-house we have a chemogenomics group. So basically that group helps us to identify small molecules, and those small molecules we check their potential of their antiviral activity in the assays with the SARS-CoV-2. And in addition to that, like I said, I've worked with a lot of biotech partners who have their drugs that they think have potential to be inhibitory against SARS-CoV-2. So we also also working with a lot of projects with other people and checking their drugs if they have a potential to stop it, because even though we have really good vaccine, we still don't have good therapeutics, and therapeutics are always needed. If someone is sick at home, you always need a good therapeutic to stop the virus infection in addition to the vaccine. So that's all ongoing. Keeping fingers crossed for this year that we get something out soon. I don't know if it's going to be preclinical, clinical, but at least hoping to get some papers out. Oh, that I'm looking at the questions like this. Another question: when you started doing the research, did you feel intimidated by it? Did you ever question your abilities? Well, yes. I mean, so my first year in grad school, like I said, I had no experience working in the lab. So, I mean, I struggled. I had to like really put in a lot of effort to figure out. The first thing that you learn about the molecular part is like doing cloning, and I couldn't clone. It was just like hard. And then I learned. I put in hours. I seeked help from like the seniors. I just find your way out, because I was just interested in the question, and I was like, I know I could do it. There's sometimes a technical challenge, and sometimes it is your own knowledge challenge. And I think at that point I was just facing the technical challenge because I never had experience working in the lab, and I thought I can never do it, but I'm here. So if I can do it, I think everyone can. And now and then you always doubt at every step. I have my lab, and I still like get some doubts. Can I do this? Can I get my people going, and can I finish this project? So you will always have doubt, but you have to fight out your fears by your confidence, and then you can do it. If you put in the work, you will get it done.